I would like to introduce the first speaker, which is Bruce Alberts. He has served as editor-in-chief of Science between 2009 and 2013, and was one of the first free United States science envoys 2009 to 2011. Bruce Alberts is a prominent biochemist with a strong commitment to improvement of science and mathematics education. Albert was awarded the National Medal of Science by President Barack Obama in 2014. He is now the Council's Leadership Chair in Biochemistry and Biophysics for Science and Education at the University of California, San Francisco, to which he returned after serving 6-year terms as the President of the National Academy of Science. Please welcome Bruce Alberts.
It's a pleasure to be here. I'm a big advocate for young academies. I wish we had one in the United States. I hope we will have one soon. I was just in Indonesia and in India this summer, both cases to try to interact with their new young academies. There's something like 24 of them now. And in many cases, the young academies are showing up, we say showing up, the old academies because they're being more active and more effective in reaching out to society. And that's only good. So I'm going to talk a little broadly about the background to this issue of how you measure science. And of course, what you measure is what you get. If Sweden measures the wrong things in judging performance of science, or if any country does it, then you get the wrong kind of science. And so it's a very important issue. I'm going to start with a little personal history because all of life is an education, and I've learned a lot from both research and from textbook writing. I've been writing textbook for over 30 years. Then what is excellent science and how can we stimulate it? And finally a little bit about how this relates to publishing. And actually I noticed in my title I had put the word teaching. If there's time, I'll say a few words about that as well. Oh, learning from research. So for about 30 years, I was a research scientist using this bacteriophage T4 that affects E. coli as a model system to study the mechanism of heredity, that is, how does DNA replicate? Make a long story short, a lot of biochemistry. We and others, over the course of about 20 years, figured out that DNA is replicated by what we call a protein machine, a complex of proteins that work much like a sewing machine, and very efficiently replicate both strands of the DNA helix. Two DNA polymerase enzymes actually catalyze the new bonds that are formed. They work at the same time, one making DNA backwards on the lagging strand because, as the famous Watson and Crick paper of 1953 showed, the two DNA strands of double helix that need to be copied are anti-parallel. They go in opposite directions, and yet the DNA polymerase molecule only goes in one direction. So this DNA polymerase can go continuously. The other one has to go backwards and make DNA in a series of short fragments. If anybody had made a theory that this is how DNA replicated back when we started, people would have said they were crazy. You know, it looks way too complicated. All this process occurs at speeds of hundreds of nucleotides per second, and the net result is only one error in 1 billion base pairs replicated. So how could you have such an error-free process with such a sloppy mechanism? But anyway, biology is more clever than scientists. So all this is just an excuse to show you a movie. It's my favorite movie because it shows the answer to something that was a mystery that I worked on for a long time. And it's produced in connection with our textbook, and so it's freely available on the web. And here we go.
Using computer animation based on molecular research, we are able to picture how DNA is replicated in living cells. You are looking at an assembly line of miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called a helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules.
So I primarily show you that movie because that's indicative of how everything works in the cell. And if it wasn't that complicated, we couldn't talk to each other. The chemistry of life, we now realize, is much more complicated than any other chemistry we know about. And we now realize that almost every process in the cell is driven by a complex of 10 or more proteins. Nature uses machines for the same reason that we use machines in our everyday life. If you organize multiple components and drive them in unidirectional ways, in our case with electricity, and in this case with the hydrolysis of nucleoside triphosphates, you could get much more efficient processing than you can by just using one protein at a time. And we also know that the same basic mechanisms due to evolution are used throughout life, and in particular the simple system that I worked on with only 7 proteins turned out to be a very good model for how humans replicate their DNA. However, as more complex organisms evolve, each function is carried out by a complex of proteins rather than one protein because it's highly regulated. And so it's easier to start often as a shortcut to understanding something about humans with a system that's analogous but simpler, more accessible. So here's some personal lessons learned from my research. Work on a mystery. The problem I worked on was obviously something important to solve. Watson and Crick predicted in general how you could transfer heredity from one generation to the next, but they had no idea of the machinery. So obviously this was an important thing to figure out. Secondly, there are remarkable homologies between living things, and I'm a big fan of model organisms like yeast and fruit flies. You'll hear more about that in a second. And finally, nearly all processes will be based on elegant mechanisms too hard to predict. You have to do experiments to decipher it. So an important challenge for the next generation of cell biologists, all the young Academy members and students, is obtaining the information needed to accurately describe the mechanism of every type of protein machine in a cell. And this will require doing the same kind of thing that we did, purify each of the components, mix them together free of other material, and see if you could get the reaction to go in a test tube so you could then dissect it mechanistically to see how it works. This is a field called protein biochemistry. I mention this because there's not enough being done today. We need new people to go work on this. So here's an exception. John Diffley's lab published a wonderful paper earlier this year reconstituting the eukaryotic DNA replication system using yeast, but the yeast system is very homologous but easier to work with than humans. And here is the summary from their paper. They have a system, instead of using 7 proteins, they needed 42 different polypeptides to get this to work. And this is a wonderful approach to really understanding how cells work, and we need to know this for many reasons, including being able to exploit our knowledge to improve human health. But this is only the tip of the iceberg. It's perhaps 1/10 of the proteins critical to DNA replication and DNA repair, so there's a lot more that we need to do. And finally, understanding the molecular details of these replication and repair pathways will be critical to improving human health. We now know that as tumors evolve, due to long periods of rare mutation, many of them become hypermutable because those cells can acquire the 10 or so mutations that you need to really escape from growth controls. And different tumors will by chance acquire a very different defect in one of these pathways. There's great potential in exploiting each particular defect to eradicate a cancer. The PARP inhibitors are a prototype which attack BRCA2 dependent defective tumors, but there are many other opportunities to do personalized medicine here if we understood more. Okay, now I'm going to switch to learning from textbook writing. This first black edition, which took us 5 years, we were in a room together, all the authors, for 365 days. So this was a 365 12-hour days, actually. And this is the starting set of authors. Jim Watson had the idea for the book. He's at the back there with a hat. And we did a lot of the writing in a house 2 blocks from Abbey Road, so that catalyzed this picture. So the 6th edition of our big book was out last December, and my major point is each time we write an edition, we are humbled by how much we still don't know. And yet students get the idea from reading textbooks that we know 95% of what there is to know because that's what we write about. I would say we know about 5% of what we need to know. And this time, for a new feature we added was what we don't know at the end of each chapter. This is not just what we don't know, but what we don't know that's really important. And to the authors, this is the most fun part of this book writing experience, thinking about what's really important. And here, these would all be, I think, very good problems to work on for scientists. So, 2 of the recent surprises of many for textbook authors, just to give you a flavor of what I mean, that we're always surprised about how complicated the chemistry is of cells. First of all, the recognition that the cell is nothing like a test tube. We always knew it had a very concentrated set of macromolecules and small molecules in a small space. But we thought, as physical chemists, that's the way I started out, collisions are so rapid you don't need to be organized. But in fact, as you saw in that protein machine, and even more broadly, scaffolds make separate biochemical compartments, even inside a bacterial cell. And that's an extensive phenomena in eukaryotic cells. In fact, Tony Hyman is working on that, giving a seminar tomorrow here, I think. So a lot to learn about these subcompartments, biochemical subcompartments in the cell, how they form, how they're reforming, how they're located. And secondly, we now know that everything's a network property in cells. Positive and negative feedback loops underlie all cell chemistry. These give rise to situations where even when you know all the interactions, the human mind can't decipher what it means. So we call those net results of these many networks of interactions emergent properties. And to understand them, we need new methods, computational methods, that not yet developed to really deal with the complexity of cellular chemistry. So my conclusion, it will probably take most of the century to gain a true understanding of how cells and organisms work. We need much more biochemistry and purified systems, but also we need new computational mathematical methods for analyzing and understanding the enormous complexity of life's chemistry. So as a biologist, there's just tremendous opportunities for new science, and we need that new science to work on important problems and be motivated by the right incentives. So how do we keep science healthy? That's, of course, part of the theme for today. Well, first of all, we must all work to stimulate innovation. And here I refer to a project that my academy, the United States Academy, carried out starting in 1993, when President Clinton came in the office, and they gave Vice President Gore the science portfolio. And he had a whole office of people working on science. And they leaked the rumor that they always leak things to see how people were going to react to before they actually officially pronounce a policy. Leaked the rumor that in order to get any grant money in the United States, you'd have to explain how your scientific findings, if you were successful, would improve humanity. What would be the practical outcome of your results? So we thought this was a terrible idea. It would drive everybody to do applied research. And we therefore produced a series of 20 pamphlets, each one 8 pages, with specific examples of how science, basic science, generates important benefits for human health in unpredictable ways. You cannot look forward, as the Vice President's office wanted us. But you could look backwards and explain how it works. And these pamphlets are still online. They're called Beyond Discovery, The Path from Research to Human Benefit. The first one we did was on the global positioning system, and each pamphlet has a centerfold with a timeline. And this is the start of the timeline for the global positioning system. We start with atomic clocks, discovered by physicists, earned a Nobel Prize, allowed you to keep time to a billionth of a second. Everybody thought that was useless, but it was wonderful discovery. But if you keep on combining it with other things in unexpected ways, you end up at the end of the timeline with a series of 24 satellites out in space, which determine where you are by exactly how long it takes the radiation to get to you, and that requires atomic clocks in the satellites. So we did that over and over again for the cure for childhood leukemia, the AIDS protease, and so on. And we'd circulate all these pamphlets to Congress, every congressional office, and we like to think we had some effect on preventing that new policy from taking effect. So how do productive ideas arise in science? Well, actually, the Academy members who prepared these 20 pamphlets learned a lot about science ourselves. We hadn't thought about it much. But when you really think about it, new science comes from combining old science in new ways. And so this is just my own work on DNA replication. Of course, we used all these other people's work and their ideas and their approaches and combined them in ways that allowed us to make original findings. So this is the fundamental reason for the explosive growth of science, because the more knowledge you have, the more ways there are to combine things, and therefore, since 100 units of knowledge can be combined in 100 times more ways than 10 units of knowledge, in principle, things move faster and faster. An obvious case is the new methods that get developed, because as we know more about various aspects of engineering and of science, we could produce new technologies that speed up. For example, genome sequencing is now costing about 1/millionth of what it cost 20 years ago. So that's a tool, new tools. But there are many other ways in which new knowledge speeds up what we do. So this is just a schematic result of my field when I was working in it, not my field today. There are many more things you could do. But there's a catch. As knowledge grows, it becomes increasingly difficult to find the right combinations, because combining the right things gets harder and harder. And this is a wonderful quote from a mathematician, French mathematician, Henri Poincaré. He said, "To create consists precisely in not making useless combinations and in making those which are useful and which are only a small minority. Invention is discernment's choice. Among chosen combinations, the most fertile will often be those formed of elements drawn from domains which are far apart. And the true work of the inventor consists in choosing among these combinations so as to eliminate the useless ones." And of course, the vast majority are useless. One thing that I see today is it's so easy to do experiments. You buy a kit and you mix something with something else and get new data. But most of those experiments should not be done. So it's very important as a scientist to think carefully about strategy before you start. And I'd just like to advertise a website that's been developed over the years by my colleague Ron Vale at UCSF. It's in the next office. It's called iBiology. Wonderful, something like 400 talks by really outstanding scientists on a variety of different subjects. A good place to get new ideas. You know, what can I learn from the way he did or she did her science that I could apply to my problem? And it's all easily accessible from your home. The major problem I see is the channeling of research topics due to training inertia. And we see this every time we write a textbook. There are areas of cell biology that nobody's working on with modern methods. And there are other areas which are vastly overcrowded where many labs are trying to beat each other out, publishing 2 weeks ahead of time. And the reason for this, of course, is that these red spaces are places where some outstanding scientists train 20 postdocs and students, and those 20 postdocs and students tend to work in the related area to what they did before, and so you start getting a very crowded space working on, say, the RAS gene or some transcription factor. Yet there's so many other places where there wasn't such a opportunity to work with outstanding scientists. We need new science, and we need, as nations and as a world, we need to find ways to get outstanding scientists to jump from the red spaces to the white spaces. Unfortunately, however, our present system strongly discourages risk-taking, and that's because basically when you want to get money from the government, you have to show that you know how to do what you want to propose in most cases. And so obviously you have to continue working on what you worked on before. And there are ways to get around that, and we need to change that. In addition, I'm a big critic, as are many others, of the U.S. National Institutes of Health, which has been overemphasizing so-called translational biomedical research, applied research, whereas basic research on biological mechanisms remains absolutely crucial for improving human health. However, if you work on cancer and publish in a cancer journal, you're going to have a big audience and a lot more citations than if you work on a fundamental mechanism in, say, a ciliate that very few people are trying to tackle, something in the white spaces. And this is part of the problem with measuring people's productivity by any automatic mechanism. So this is 250 editorials I had over 5 years of scientists, one a week. This is my favorite one. It's written by Huda Zogby. She's a MD, never got a PhD. She was originally from Lebanon, and she came to the United States, and she was treating young children with Rett syndrome, and she realized she could do nothing for these children, so she started to learn how to do research, and now she's a great translational researcher at Baylor. But she wrote this editorial complaining about how everybody is getting pushed away from doing the fundamental basic research that she needs to think more effectively about her own problems. And the wonderful quote from this is, "The challenge in translational medicine is that scientists are trying to translate a text with the sophistication and depth of Shakespeare using a first grader's vocabulary and experience." That's such a wonderful quote. And only somebody like her who grew up speaking Arabic and then had to learn English would think of this. but it's such a powerful editorial, especially coming from a person with her interest. So just one example, the importance of the fruit fly. I could give this in many different examples. We're still far from understanding how cells work together to form and maintain tissues. Many examples show that at first working on a mechanism in the fruit fly Drosophila provides a shortcut to understanding humans, a faster way than going directly at humans. And in our chapter on the development of tissues in this blue book I just showed you, we have 50 references to Drosophila, 4 times more than the next most cited organism, which was the mouse. So the problem, this is what's happening in the United States. This is an analysis recently published in the journal Genetics. In the last 5 years, there's been a 30% drop in government support for fruit fly research compared to only a 15% drop overall. It's the kind of thing we see all the time. Young people feel they're forced to work on human cells or mouse cells. To understand cells, we also need new models. This is something I like to promote. This is from a young assistant professor at UCSF, Wallace Marshall, who his whole research career had been working on cilia in Chlamydomonas. And he made a leap into one of those white spaces very courageously to go back to study this mystery about how cells, eukaryotic cells, know how to pattern their surface. Ciliates have an enormously patterned surface, and this ciliate stentor was extensively studied by classical biologists, including T.H. Morgan before he worked on Drosophila. And you could cut them up in little pieces and paste them back together, and they regenerate. This is all one cell, though. And so they have like a GPS system on their surface. Now, I will bet that human cells also have similar kinds of abilities to know where things are on their surface. But this is the place that seems to be a very good place to try to work that out. But yet, nobody was working on that. This is his first published paper. And there's nobody else working on it. We hope we get more people working on it. He's not going to have a lot of citations, and it took him a long time to get this paper because he had to start from scratch. So anything that's measuring number of publications or citations is not going to encourage creative research like this. So this is what I just said. I hear this. Everybody tells me I won't be able to get a research grant from the U.S. National Institutes of Health unless I shift to work on mouse or human proteins after my postdoc. So this kind of thing led four of us, Mark Kirshner, Sherry Tillman, and Harold Varmus and I, to publish this PNAS paper in April 2014 called Rescuing U.S. Biomedical Research from Its Systemic Flaws. Sherry Tillman was a former molecular biologist, just stepped down as president of Princeton. Harold Varmus, former NIH director, just stepped down as head of the National Cancer Institute, and Mark Kirshner is the head of the Department of Systems Biology at Harvard. This article has got a lot of attention. People have been basically afraid to criticize anything in our system, fearing that if we point out that we need to change something, maybe the politicians will just take away more money because we're not doing everything perfectly. It's like, it's a very bad system. It's like, you know, Soviet Union or China or something like that. You can't fix things if you don't know about them. So we've been trying to launch a movement to get movement on this issue. I can only highlight a few things. One of the things that is very appropriate for the Young Academy, and I think there's been a disturbing change in the age distribution of independent researchers in the last 30 years. This is in the United States. Somebody has to get the data for Sweden. This is a nice article by the president of Johns Hopkins that followed ours. He points out that the proportion of all grant funding awarded to scientists under the age of 36 has dropped from 5.6% of the total budget of the National Institutes of Health in 1980 to 1.3% in 2012. This is horrible. Here's a question. How successful would Silicon Valley be if nearly 99% of all investments went to innovators who were 36 years old or older? I think we'd be dead. So specific recommendations, there should be many more grants for young investigators that encourage and demand creativity and risk-taking. And secondly, agencies should remain sensitive to the scale of funding of individual laboratories and should regularly review and sunset large-scale expensive projects which tend to go on forever because they get a constituency. Large-scale expensive projects, programs, and intergroup efforts which in general are often not productive after a while, but yet go on. So here's a specific proposal. This is really, Tony Hyman and I are going to work on this, preparing more details of this, but focus on generously finding the best young investigators within 4 years of their PhD with the aim of replacing the 2,000 independent investigators aged 36 or younger who have been lost since the 1980s. There's now fewer than 600 of these people. There used to be 2,600. We want to encourage ambitious aims and do not require preliminary results. And we want to review these applications using broad groups of outstanding investigators to avoid the siloing effects of the current review process where, you know, the radiation biologists look for good radiation biology. And we want to look much more broadly than that. So now we get to the central focus, basically, of this workshop, that basically scientists are actively discouraged from innovative research that departs too far from their previous work, partly because of the reason that I just said. If you're going to start something new, you're not going to publish much for a while. So any evaluation must weight research quality much more heavily than quantity. For example, many places now in the United States are specifying that a small number of publications be emphasized on grant applications and job applications with the expectations that if you only give a few things, the reviewers will actually be able to read the papers. If you give a whole list, they'll just look for where it was published. And we must stop using lab size or total funding as any measure of real success in science. You know, there are many countries that, like China seems to want to have the biggest lab, the most money, that's the most success. This is not the way to get good science done. This is my first editorial, How I Got into Science Policy, 1985, Limits to Growth in Biology, Small Science is Good Science. So I've been on this trend for a long time. This editorial in Cell was severely criticized by lots of people in follow-up editorials who had big labs. So, publishing scientific research today. The search for automatic ways to evaluate science is distorting how science is done. And the worst of these is the evaluation of an individual scientist by the impact factor of the journal in which his or her research is published. And you'll hear a lot more about that from speakers today. What is a journal's impact factor? The impact factor was developed by a scientist, Eugene Garfield. He wrote a nice article on the history and meaning of the journal impact factor years afterwards. And he points out this new tool was introduced to help librarians decide which journals to purchase, not to evaluate scientists. And a journal's impact factor is based on two elements. The numerator is the number of citations in the current year to items published in the previous 2 years. So how many citations in the last year to anything published in the previous 2 years? That's then divided by the denominator, which is the number of, quote, substantive articles and reviews published in the same 2 years, and that's part of the problem. But anyway, so basically, you get a number, and the numbers now produced by a company called Thomson Reuters. Garfield sold the rights to that company, and many of us in 2012 got together to produce, in response, the San Francisco Declaration on Research Assessment. And the basic recommendation of this petition, now signed by something like 10,000 groups and people, is do not use journal-based metrics such as journal impact factors as a surrogate measure of quality of individual research articles to assess an individual scientist's contributions or in hiring, promotion, or funding decisions. So this petition has been widely known, but it's one thing to say something, it's another thing to get it done. This is an example of how you start to get it done. This is from Sandy Schmidt, she's chair of the Biochemistry department at UT Texas Southwestern. Here's how they announce their new jobs. Importantly, we reject the notion, this is in a science magazine or Nature where they're advertising their position. Importantly, we reject the notion of where you publish or where you graduated from defines the quality and impact of your work, or is necessarily predictive of your potential for future success. Rather, we believe the best candidates for these positions will be able to provide succinct and compelling answers to the following 4 questions in their 1 to 2 page cover letters. 1, what is your most significant scientific accomplishment as a graduate student? 2, what is or will be your most significant accomplishment as a postdoctoral fellow? 3, what will be the major focus of your independent research program? And 4, how will your future research benefit from and be a benefit to UT Southwestern? So the applicant has to actually see what's happening at UT Southwestern and write something intelligent about what they think. This last part takes work. So impact, as a former editor of Science, I can tell you impact factors are also distorting how journals behave, even the best journals. Science the people who sell ads, the business office at AAAS, who runs Science, want the highest impact factor as possible because that gets more ads. Because they have they think the advertisers will want to publish in Science more if it has a higher impact factor than Nature. So that puts pressure on journals to game the system. And I know that every journal, almost every journal is thinking about this. We could easily raise your impact factor by publishing more reviews, because reviews are always cited more than papers, and they go into the numerator. Or by publishing less research in less populated areas of science. So when I was editor-in-chief of Science, we started publishing education research. We're not publishing much, since I left, they're not publishing much education research. I think part of the reason is they're not very well cited. So that's going to lower your impact factor. So you want to publish everything on cancer if you want to have a high impact factor, but it's not going to produce the best journal. Oops, I'm wrong computer. So we had a follow-up meeting 2 years later after the DORA with some of the same people, with the actual people at Thomson Reuters who produces impact factor, and we made some simple requests, which they're still considering. First of all, provide separate journal impact factors for reviews and for primary research, so that journals won't feel compelled to publish all these reviews, and it will have some more meaningful number. And for each journal, provide the median value of citations per citable article rather than the mean. When you look at the actual way impact factors are determined, 1 or 2 papers can get 10,000 citations and completely change your impact factor. So how is that related to the quality of the journal? And then stop quoting the impact factor to 3 decimal places. It's, at best it's accurate to 1 decimal point place. It's crazy. It's completely non-scientific, but it allows them to rank journals give a real ranking. That's the only reason why they do it, I think. But that ranking is fallacious. Okay, so I just want to end by this question you'll hear a lot more about. Can a major move to preprint servers help? There's been a bias in biology not to use preprint cycles. You'll hear later today about physics has been really a pioneer here. There is something called, several preprint servers, one's called BioArchive, and Ron Vail, the same guy who did the iBiology website, is a fantastic scientist. He wrote an article called Accelerating Scientific Publication in Biology, which is now in press in PNAS, but was posted right away on the archive server called BioArchive. And at any rate, I just want to give you a quote from his article. Preprints could benefit scientists if they first allow them to establish priority for discovery in a more predictable way than navigating an unpredictable journal review process. 2, allow them to use preprints as evidence of productivity in grant applications, particularly in cases where a new research direction is being pursued. 3, enable graduate students to provide evidence of scholarly work for graduation or postdoc applications, thereby potentially decreasing their training time by many months. Because he points out to get a paper published takes often longer than your whole normal graduate career. And 4, allow scientists to obtain feedback on their work earlier than is currently possible through the journal system. He has a whole discussion of how this might prevent some of these high-profile, fraudulent, incorrect papers from getting actually published. And it will take a strong push from young scientists to generate change. So this is why I always go when I'm invited by young academies. This is a paper written by 600 postdocs who have a meeting every year in Boston. This is called Shaping the Future of Research, a Perspective from Junior Scientists. It was published in F1000 Research. And part of the effort of older folks like me has got to be giving a larger voice to the next generation of scientists. And so this gang of 4 that started this process, me, Mark Kirshner, Shirley Tillman, and Hera Varmus, has now been expanded to 16 people, and I'm just showing you some of them. 2 of them are very young. Jessica Polk is a postdoc at Harvard, and Daniel Cullen-Ramos is an assistant professor at Yale. Very active people in the, and Jessica was one of the organizers of that big postdoc meeting in Boston. What we've done is generating a website called Rescuing Biomedical Research. I should point out that Tony Hyman is also one of these 16 people. There's the steering committee is listed here. And the basic idea is to, for example, after this session today, Tony and I are going to try to work on a proposal for young scientists, how we could try to get those 2,000 young scientists back as principal investigators. We're going to have to write up something pretty good to convince the other 14 members that this is a good idea. And then the steering committee votes, and if it gets, I think, 11 out of the 16 votes, then it goes up on the website under progress. You know, this is what we're advocating for. And we're going to try to tie those things into the people who could make a difference, the Association of American Universities, the National Institutes of Health, and so on. Anyway, this is our attempt to, and very importantly, we're trying in this way also to give a much louder voice to what young people think than the voice they might normally have. So I was going to, my title has on science education, but I'm out of time. I just want to not forget to make the point that one of our big issues today is changing the way we teach science at all levels, from schools, active science, to universities. This is my favorite example of a university that's been really pioneering. University of Minnesota, they built a science building with 16 flat lecture rooms that each one accommodates about 200 people around these tables of seat 9 people with 2 computers. And this is active learning in college biology class. They're teaching all their first-year biology students, thousands of them, first-year biology in this way. Basically, this is the only way we're going to get change at lower levels is if the people at college with all the prestige start defining a science education in a different way from just talking at people and telling them facts. There's a new publication from the National Academies in the United States that I highly recommend. Free PDF downloads from the National Academy Prep's website called Reaching Students, What Research Says About Effective Instruction in College, Undergraduate Science and Engineering. It's a wonderful book that every professor should actually have on their reading list to try to encourage them to experiment with new things and new ways of teaching in their classes. And this is my favorite quote from Jacob Bronowski, Science and Human Values. Physicists who saw the atomic bomb results in Hiroshima and Nagasaki in Japan shortly after the war. He was flown over that area as a a member of the British Army, and he got very depressed, and he spent 10 years reading and writing about whether science was good or bad for the world, and he concluded, of course, that it was good for the world. He said the society of scientists is simple because it has a directing purpose to explore the truth. Nevertheless, it has to solve the problem of every society, which is to find a compromise between the individual and the group. It must encourage the single scientist to be independent and the body of scientists to be tolerant. From these basic conditions which form the prime values, there follows step by step a range of values: dissent, freedom of thought and speech, justice, honor, human dignity, and self-respect. Science has humanized our values. Men have asked for freedom, justice, and respect, precisely as the scientific spirit has spread among them. So my talk today has been focused on the United States, but I've given versions of this talk all around the world. I'm just in Brazil, just in Indonesia, India. Afterwards, students come up and say, we have the same problem here. You know, our education is learning science by rote. We have, young scientists cannot be independent. We got to work on everything that we worked on before. I hear it all over the place. I would not be surprised if Sweden also has the same problem. So this is the last wonderful quote from Louis Pasteur. Thank you.
Thank you, Bruce Alberts, and I wonder if anyone has any question in the audience. Now it's the time.
Thank you, Bruce. Great, wonderful talk, Bruce. So you talk here a lot about things, how we use journals to publish, and of course, open access journals or even preprint service is an amazing thing there. But another aspect is, of course, what are the journals' responsibility for making sure that some research is either not published or even pulled? You could argue that Andrew Wakefield and the vaccine scandals is one example, right? Mm-hmm. Could this become a problem with increasing use of open access archives? That anything can be put online and can't really be pulled?
Well, yeah, I'm sure this will come up later today, yeah. Ron makes the point that if we had preprint servers that really worked, they would get a lot of eyes on papers early, and his hope is that there'd be less irreproducible results actually published. He says at the end, am I sure that this is a good idea? He said, no, but let's do an experiment. Anyway, I am extremely concerned about the image of science now with so much pressure to publish things that are wrong. People are publishing things in biomedical fields that, sound good, and in my opinion are not fraud, but involve highly selected data, because you could imagine, a postdoc or graduate student, the only way to be successful is to find the result that the lab has been working on to be correct. I actually teach a course still teaching with Cynthia Kenyon called Classical Papers in Biology, and it's the 10 sessions, 3 hours a day, and basically we always pair a classical paper like Darwin, Darwin's always looking for what could be wrong with what he's saying. We pair that particular one with a paper high-profile paper that's wrong. It had 20 authors from Harvard and MIT. So what's going on here in science? And I'm extremely concerned that a lot of things are incorrect in the literature. They never get corrected, really. People know they're wrong, who are in the field, but most people don't know. And in fact, you find that in teaching this course, we find that the incorrect paper, which was refuted, and everybody knows it's wrong, but was refuted 2 years later, still last year, the incorrect paper was cited maybe 5 times more than the incorrect paper. So we need to do something. And there's so much literature out there, and people, I suspect that people's reputation, it should be that your reputation is improved if you point out what was wrong with your old papers. But right now, I think your reputation improved if you ignore that your old paper was wrong and just move on. And so we have major problems to solve. And I don't know what the answer is, but I'm very concerned.
So you have this list of 3 ways that Thomson Reuters can improve the impact factor. Is that because you think that the impact factor is a good way for judging scientists?
No. I think the impact factor as it stands is distorting what journals do. So at least that effect would be improved if you separate reviews from primary. If you, and of course, 3 decimal points is absolutely crazy. It's so non-scientific. But you'll hear a lot more about it. So should we just let it go on as it is, not try to get them to improve it, or should we try to improve it? It's sort of an enemy, but it's such a badly done enemy right now. You at least like to see them being thoughtful about it.
Yeah, but my worry would be that if they improve it, that could be used as an argument.
Okay. It still won't be a measure of individual papers.
Thank you, Bruce Alberts. And I want...